Shall
Okay, very good afternoon, my dear friends. Today's one of the eminent resource person, Dr.K.MADHURI Madam Garu, Assistant Professor, Department of Botany and Biotechnology, one of the prestigious institution, that is Mahatma Gandhi University in Nalgonda. Madam is going to explain one of the important area in molecular biology. So, my dear students, listen carefully. Madam, please start your session and share your screen.
Okay, sir. I'll start sharing the screen, sir. You please let me know. Hope everyone is able to see my screen, sir.
Yes, madam. Yes. Please click on slideshow.
Yes, sir. I'll do that. Yeah. So I have chosen this topic, the nucleic acids, for today's session. That's a basic one which every student go wrong about the DNA and the RNA and the composition of the DNA and the RNA. So to give more clarification on this, I have chosen this particular topic where we will try to deal with every molecule which composes of the nucleic acids. So as we know very well that Friedrich Mischer, he's the first person in 1869. He's the first scientist who has isolated this particular nucleic acid, which we know today. He has isolated this as nucleon from the nucleus of a pus cell. Since this particular nucleon is in a kind of acidic in property, later it was named as nucleic acid. So as you can see, majorly we study about two types of nucleic acids. One is deoxyribonucleic acid and the other is ribonucleic acid, where the deoxyribonucleic acid and the ribonucleic acid, majorly the difference falls under the sugar molecule. So as we all know that the nucleic acids in eukaryotes specifically, they are embedded to a particular membrane, the nuclear membrane, whereas in prokaryotes, they are distributed throughout the cell. I mean, they don't have confined nucleus in prokaryotes. Whereas in eukaryotes, you have DNA, which is found in the form of a proper nucleus. And this nucleus is in a circular maximum, and it is covered with, your nucleus is covered with nuclear membrane. And this DNA is found in the nucleus with small amounts in mitochondria as well as in the chloroplast. That's why we differentiate and study the DNA which is present in, that is mitochondrial DNA and chloroplast DNA. Whereas RNA, this is found throughout the cell, and we majorly study three types of RNA. But today we will concentrate on the DNA structure followed by the RNA structure. As you can see on the screen, this DNA we generally study as a genetic material, and it is present in all the cells, virtually restricted to the nucleus only. And as you can see, the amount of this DNA in somatic cells are the vegetative cells or non-reproductive cells, which we all know. And of any given species, it is generally constant. The amount of DNA is constant. If we try to take the DNA of a particular cell, especially of a human cell, our length of the DNA is around 2 meters. Just imagine a cell is not like, it's a microbial cell. Like, I mean, we generally observe the cell under the microscope. Whereas in this cell, the DNA which is present in the human cell is 2 meters in length. So just imagine the miracle behind the DNA. So the amount of DNA in somatic cell of any given species is constant, like the number of chromosomes. Like we very well know what pairs we have and all. So the DNA content in gametes are the reproductive cells. So generally they have half that of the somatic cells. And in polyploidy, like we know that we generally take the DNA or the chromosomes in sets. So if it is more than one set of chromosome, then it is called as polyploidy, where we generally study about diploid, triploid, tetraploid. So three types. So those are the polyploidy. So in cases of polyploidy, the DNA content increases by a proportional factor. And if we take the mutagenic effect of UV, like if we generally subject the DNA under UV light, the peak, the maximum peak, which goes beyond is 253.7 nanometers. So the peak for the absorption of UV light by DNA is 253.7 nano. We can see the DNA under this particular frequency. So as I mentioned you, the nucleic acid, if we take the nucleic acid structure, your nucleic acids are polynucleotides. That is, they are the building blocks of nucleotides. So from here, I have found a very good PPT, which I'm going to share with you guys now. So in this PPT, it is more clear about the, yeah. So as you can see, we have two types of DNAs, that is the nucleic acids, that is deoxyribonucleic acid and ribonucleic acid. And as we mentioned, the DNA is found in nucleus with small amounts in mitochondria, chloroplasts, and RNA is found throughout the cells. So we mentioned that these nucleic acids are polynucleotides. They are the building blocks of nucleotides. So let us see what are these nucleotides. So as you can see that, like how our house is built up with bricks, the same way. Now, only bricks will not be the picture, but the bricks play the major framework of the house. The same way in DNA, the nucleotides act like bricks. They are the energy-rich compounds and which drive the metabolic process in cell. Whatever the metabolic process, like the Krebs cycle, whatever the cycle, metabolic pathways which the cell has to undergo, this is driven with the help of the nucleotides. And these nucleotides, they serve as chemical signals, like cell signaling we say, right? So this is possible by the metabolic process, which is being in turn taken by the nucleotide. So they serve as a chemical signals and key links in cellular system that respond to hormones and other extracellular stimuli. So these extracellular stimuli, hormones, these all we require to trigger a particular pathway. So this is possible by these building blocks. And as you can see, they are structural component of number of enzymes, cofactors, and metabolic intermediates. And each nucleotide is formed by basically 3 units. The whole nucleotide is composed of. Now, as I mentioned you, the nucleotide is a building block, like the brick of the house. Now, this nucleotide, definitely the bricks are supported with cement, soil, right? The same way this nucleotide is formed with the help of phosphate, sugar, a phosphate group, a sugar group, and a nitrogenous base. So as you can see here very clearly, so a nucleotide is made up of a phosphate, a sugar. A sugar group is what? In RNA, the ribose sugar is present. In DNA, the deoxyribose is present. And the bases, these are the nitrogenous bases which form the nucleotide. You can either have a purine base and a pyrimidine base. Your purines, generally there are 2 types, that is adenine and guanine. Whereas a pyrimidine, you have 3 types, that is cytosine, thymine, and uracil. Uracil comes into the picture when RNA is there, and cytosine and thymine come into the picture when DNA is there. And this nucleotide without the phosphate group is called as nucleoside. That is, only the sugar and the base together is called as nucleoside. Why side? Presence of sugar, absence of phosphate. Whereas if phosphate group is also present, it is called as nucleotide. Hope I'm clear with it. So let us see the phosphoric group or the phosphorus group which is present. And this molecular formula is H3PO4. And basically this contains, as you can see, 3 monovalent hydroxyl group, OH, 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 and a divalent double bond oxygen atom. And they all link to pentavalent phosphorus atom. So P with 1, 2, 3, 4, 5 bonds. So it's a pentavalent phosphorus group. And coming to the sugars, you have the sugar structure is made up of 5 carbon keto sugar or pentose. 5 carbons are present. And each carbon, they possess D to deoxyribose and other content D ribose. That is deoxyribose in DNA and ribose in RNA. And these sugars are present in furanose form and beta configuration. That is a structural form of the sugar. And as you can see, the pentose sugar form esters with phosphoric group. So to the sugar, you have the phosphoric group attached, and this is called as phosphodiester bound. Let us see the structure of it over here. Here it is a nucleoside. I will give you the clear picture how the sugar and the phosphate form the phosphodiester bond also. Now let us see the nitrogenous bases. As I mentioned, we have two types of nitrogenous bases, that is purines and pyrimidines. You have purine derivatives and pyrimidine derivatives. So this base is linked to the sugar. The purines and pyrimidines are linked to the sugar with the sugar-sugar bound. And as you can see, the purines which are present in DNA and RNA are adenine and guanine. Whereas in the pyrimidine derivatives which are present are uracil, thymine, and cytosine. So here comes a nucleoside. As I said you, nucleoside is the sugar with the nitrogenous base. There is no phosphate group attached. So when ribose or deoxyribose is combined, that is the sugar group. If the sugar is ribose, it is RNA. If the sugar group is 2-deoxy, it is DNA. So if the ribose or the deoxyribose is combined with purine or pyrimidine base, it is called as a nucleoside form. So without the phosphate group, it is nucleoside. If phosphate group is present, it is called as a nucleotide. So as you can see here, you have a beta glycosidic bond between the D-ribose and a cytosine nitrogenous base over here. This is the bond you can see. This is the nitrogenous base, and this is the sugar molecule where it is forming the nucleoside. Now you can see the difference. As we mentioned, two types of sugars, ribose sugar, deoxyribose. So in ribose, as you can see, there is a oxygen missing over here. So we count here like 1, 2, 3, 4, 5. 5 carbon, right? 5 carbon compound. So 1, 2, as you can see here, hydroxyl, OH is hydroxyl. So in ribose, hydroxyl group is present, whereas in deoxy, that is in the second group of your carbon, you have only hydrogen present. Okay. So there's an oxygen missing. So this is the major difference between the ribose and the deoxy. You can see here, 1 OH, H. Then coming here, 1 H, 1 OH. Here OH, OH. And here you can see CH2OH and H over here. Whereas over here, only 1 H is present. And as you can see, this sugar phosphate, the sugar and the phosphate. Here we are not showing the nitrogenous bases. This is only the sugar group along with the phosphate. This is called the backbone of the DNA. So whatever the DNA or RNA you see, the backbone of the DNA will be a sugar and a phosphate bond. So I mean a phosphate group. As you can see that this is the ester bonding. The sugar and the phosphate go for the ester bonding. So the nucleotides are also oriented in the same direction, one after the other. Nucleotide means what? A carbon, a phosphate, and a nitrogenous base. So as you can see, the phosphate groups joins the third carbon. You can see here the phosphate groups over here. It will join to the third. Okay, here you can see. Okay, it will join to the third carbon. As I mentioned you, 1, 2, 3, right? So the phosphate groups joins to the third carbon of one sugar and to the fifth carbon of the next line sugar. So the phosphate is between the 3 and the fifth one. So as you go on, here comes the adding of the bases. So now the phosphate is towards the third carbon and the fifth carbon, whereas the nitrogenous bases are added to the first carbon of the sugar group. So the order is important to determine the genetic information of the molecule. This order is very much, it has to be maintained to determine the genetic information of the molecule. And as you can see that deoxyribonucleic acid, it is present in every living organism, and generally even the plant possesses and the animal possesses the DNA, but the organization is different. That's the reason the genes which are present, they trigger and they try to give a different character. So as you can see, this deoxyribonucleic acid, this is a material of inheritance. That is, it is been transferred from one generation to the other generation, and it was discovered in 1960 by Franklin, Watson, and Crick. They gave a particular model for the DNA today, which we study as Watson and Crick model. And this Franklin, she's a lady who is behind the the structure of DNA. And through series of experiments, they have concluded that the DNA is a genetic material which is present in the nucleus of the cell. They have conducted n number of experiments stating that this is the DNA which has been inherited from one cell to the other cell, and one generation to the other generation. And as you can see that the human DNA contains almost 3 million deoxyribonucleotides residue and containing around 25,000 genes. As I mentioned you again, the DNA of one cell of a human being, if we take and measure, it comes up to 2 meters of length. So these genes, they are stretched on the DNA that carries the codes of protein production. So these genes are the one which code for a particular protein, which code these proteins help in the metabolic activities of the cell. So as you can see, the DNA, it is a polynucleotide chain. You can see so many nucleotides, that is phosphate, sugar, your nitrogenous base. So you can see the building blocks, and this forms a polynucleotide. And generally the DNA exists in a 2-strand polynucleotide chain. And here you can see that here the phosphate group is in the 5th carbon. So this chain is running from 5' to 3', we say. And here the phosphate is in the 3rd carbon. So hence it is called 3' to 5'. So you will study as we go ahead. And you can see that the DNA is generally made up of 2 strands of polynucleotide. And these strands, like generally we take it as a lagging strand and a logging strand. And the sister strands of the DNA molecule runs in opposite direction. You can see that it is running in opposite direction. So this is in one direction, this is in another direction. So they run parallel, anti-parallel to each other. And they are generally, the 2 strands are joined generally by the bases. And as you can see, each base, you have the nitrogenous bases as adenine, guanine, tyrosine, and your cytosine, right? So always the ATGC pairing takes place. That is always, as you have seen here, that so here we have seen the purines. The adenine always binds with the thymine, ATGC. So the adenine binds with thymine and guanine binds with cytosine. That's why ATGC binding we say. Adenine with thymine, guanine with cytosine. And generally the adenine binds with two bonds. You can see that AT is always with 2 and GC with 3. So that's why this is the stronger bond when compared to AT. Okay, so here comes your A always pairs with T and G always pairs with C. And as you can see, always a purine is paired with the pyrimidine. There is no purine or pyrimidine. You have a purine pairing with pyrimidine. So ATGC, and thus the sister strands are complementary but not identical. So if in one strand, if it is A, the second strand will be T. And in the first strand, if it is G, the second strand will be C. So in this particular strands, you can see if there is G, your complementary strand will be C. And in the parent strand, if it is C, then the complement will be G. So as we are saying, they will be anti-parallel to each other, and they are bonded by hydrogen bonds. These are hydrogen bonds. So A always bind with T with 2 hydrogen bonds, and G always binds with C with 3 hydrogen bonds. Now, if I say a question, students, that if like in which particular DNA the hydrogen bonds are more, means the DNA which is having GC content, more of GC content, definitely there will be more number of hydrogen bonds. So here are the purines, the adenine and the thymine. So adenine always binds with thymine and guanine always binds with cytosine. So ATGC purines with pyrimidine bonding. So it is a single ring structure, and here comes a double ring structure. This is Watson and Crick model of base pairing. So these two scientists are given the complementary structure of the DNA, how the first strand binds with the second strand, forming a complementary binding DNA structure. So here you can see the thymine sugar, the nitrogenous base, the thymine nitrogen binding to the adenine. Okay, and here comes the cytosine binding to the guanine. You can see two hydrogen bonds between T and A, that is AT, two hydrogen bonds, whereas in guanine you have three hydrogen bonds. So the DNA containing more number of GC content will have greater hydrogen bonds. These are the two great scientists, Watson and Crick, who gave the double helical structure of the DNA in 1950. They were the Nobel laureates. And as you can see that this blue is one strand and the pink one is the complementary to the blue strand. That is, if this particular strand has A, then this pink strand or the reddish strand, they will have T. So A, T, G, C is the complement. So this is the basic structure of the DNA. If we understand, this DNA structure can be understood in three levels. That is primary structure, secondary structure, and tertiary structure. So this primary structure, basically, it involves the sequence of its nucleotides, how the nucleotides are arranged. So the nucleotide residue form the primary structure, and the secondary structure gives the how the helix has been formed between the two DNA strands, and the third structure, this gives the dimensional shape. That is, we generally say 3D dimensional, right? 3D structure. 3D structure is nothing but three-dimensional shape. The three as what is the 3D? Like you can see in the three sides of a particular structure. And this arises from the supercoiling, where double helix is being twisted into compact shape. As I mentioned again, the cell contains 2 meters of DNA in human cell. Means just imagine how the DNA is compactly arranged in one tiny or one micro cell of our human being. So here in the next slide, if we see the DNA replication, this DNA replication is basically carried in all the information which is required for us is been making the protein required by living organisms. So what is a DNA? So as I mentioned, it is building blocks of nucleotides. These nucleotides, they help in bringing up the metabolic processes, and these metabolic pathways, how they are built, how they are done. So in simple, we constitute it as the DNA carries the information which is required for making the protein or making a particular metabolite required by the living organisms. And of course, we also mentioned that whatever the information, to pass information stored in DNA to next or the new generation of the cell, the DNA has to replicate. And if it replicates only, replication is what? Multiplication. If the DNA will multiply, then only the DNA will be doubled, and that double DNA will be transformed to the next generative cells. So the DNA, the genetic material, has to be passed from one cell to the other cell. So the information has to be carried from one cell to the other cell. So this is possible only by DNA replication. And as you can see, this information is carried in primary structure of DNA, that is a nucleotide. And this, to pass the information stored in DNA to a new generation of the cells, DNA replication, as we mentioned, it is must to be taken. And when DNA is replicated, each strand of the double helix serves as a template. What do you mean by a template? A template is the information which we try to copy onto the next strand, and this next strand is being transferred to the next cell or the next generation. So template is a kind of a copy, what I should say, a prime copy of the information which is already existing. Ante like this copy of the information which is there in the parent, that we take it as a template and we manufacture the new strand of DNA. It's like a kind of a master copy, okay, which is existing in the parent cell. From the master copy, we are trying to replicate and prepare few more copies, and these copies will be transferred to the next generation. And as you can see, in each of the daughter DNA strand, one strand from the parent DNA is present. So it replicates, and the replicated DNA is being transferred to the parent, to the daughter cell. And this in this daughter cell, further they replicate, and so on. The DNA is being replicated from one cell to the other cell. And generally, the DNA from the template which we copy, and after that which is being transferred to the daughter cell, is called as a semi-conservative replication, because half the replicated copy will be transferred to the daughter. So that's why it is semi. And the production of new DNA is carried out by enzymes called as DNA polymerases. We have the enzymes, 3 types of enzymes actually. The majorly 3 types. There are many, but majorly we study 3 types of enzymes, and those we classify under DNA polymerases. And these DNA polymerases, they catalyzes the addition of deoxyribonucleotide residue to a growing DNA strand. So as you can see that the parent, this is a kind of a semi-conservative replication which we are going to see now. So here you can see the parent molecule which is having 2 complement strands of DNA, and each base is paired by the hydrogen bond with its specific partner, like A with T, C with G. So these are the 2 complementary strands which are present in the parent. Now, if they have to replicate, what will happen? You can see they get separated, and in the first step, the replication is separation of 2 DNA strands. So the bonded DNA, they get separated. Now, each separated DNA, now they serve as template. Like from this original copy, we are trying to make a duplicate. So from this template, we are trying to make another copy of the nucleotides. So you can see the dark blue one represent the parent strand, and they serve as a template. And this determines the nucleotides which has to come on to the complementary strand. So as you can see that in this one, you have A, C, T, A, G. So definitely the complement will be T, G, A, T, C. Whereas in the other strand, it is T, G, A, T, C. And the complement for it will be A, C, T, A, G. So you can see that A, T, G, C, A, T, A, T, G, C combinations. So in this way, you can see that the nucleotides are connected to form. So they are being again bonded with this particular template strands with sugar-phosphate backbones of the new strand, and each daughter DNA molecule consists of 1 parental strand and 1 new strand. So this one will go to the daughter cell. So when this goes to the daughter cell, you can see that the daughter cell will have 1 parental strand and 1 the newly formed strand. So this is called as semi-conservative replication. So let us see how this is carried out. Sir, do we have time? Shall I continue here or shall I stop it? Sir, Tirupati sir, shall I continue the replication?
Madam, only 9 minutes time is left. Only 9 minutes. Please
Okay,
proceed.
so,
only 9 minutes.
Okay, sir. I'll start off the replication part now. In the next session, I will continue the replication part. So as you
Yes,
can
madam.
see, as you can see here, your parental DNA is from, you can see here, Now this is the one, you can see from 5' to 3' it is running. And here it will be from 3' to 5'. So 3', as I mentioned, the phosphate group at 3' is stopped. And here at the 5' end of that is the carbon, on the carbon, these particular phosphate groups are bonded. So yes, in DNA, this is the picture which is showing you a detailed picture how the replication is taking place. The semi-conservative replication is taking place. So in this, you can see that the DNA is getting unwinded. So the DNA first, it will get unwinded and it will be forming a fork kind structure. So this is called as a replication fork. You can see it is in the kind of a fork structure. And in this, as I mentioned you have a leading strand and a lagging strand. The two template strands of the parental DNA is further divided into a leading strand and a lagging strand. So the leading strand is the one which will have the complementary DNA strand without any disturbance, and it is a proper strand which will be leading the genetic information from one generation to the other generation. Whereas the lagging strand, it is having little complications by formation of this Okazaki fragments, and there is a kind of informational disturbance. So that's why we generally call it as a lag. They lag the information. That's why it is a lagging strand. So let us see in depth about the replication, like why leading, why lagging, and what are the important components which is required for the replication then in the next session. But for your knowledge for today, kindly you can see that your leading strand, the part, I mean, if you see the structure of the translation is what? Passing off the information from one to the other. So how is the information passed? I think you all have an idea of central dogma, right? The DNA, the information passed from the DNA to the RNA and then the protein is called as a central dogma. So in replication, we are trying to pass the information from one DNA to the next daughter cell DNA by semi-conservative replication. So in the semi-conservative replication, you can see that you have a 5' to 3', 3' to 5' DNA. And as you can see that the 5', the light blue color strand over here, this is your leading strand. Generally, the 5' to 3' is called as a leading strand, and the 3' to 5' strand is called as a lagging strand. So here, the leading strand will be acting as a template, and you can see that you have certain enzymes which involved in replication. Those enzymes, as we already mentioned, DNA polymerase, and you have three types of DNA polymerases, that is polymerase 1, 2, 3, and other than that, the enzymes involved are topoisomerase, helicase, RNA primase. So these are the major enzymes which are involved in DNA replication. So coming to the RNA, as you can see, RNA, it is very much similar like to DNA. And this is a long unbranched macromolecule. And generally the nucleotides join from 3' to 5' end as with the help of, by forming phosphodiester bonds. And as we mentioned, RNA, you have ribose sugar. DNA, we have deoxyribose sugar. And the number of RNAs, that is ribonucleotides in RNA, range from as few as 75 to many thousands. And as you can see that this is a structure of RNA. This is very much similar to DNA. They generally change at the sugar, and both sugar phosphate polymers and both have nitrogen containing bases attached. The major difference over here with RNA is it contains monosaccharide residue and it contains a basis, that is you have ATGC, right? So here you have AUGC. Instead of thymine, your uracil group come and binds with the A. So instead of AT, it is AUGC. And the RNA exists as a single strand instead of double strand. Majorly, the RNA exists as single strand. So here you can see that these are the nucleobases and the base pairs. This is in the supercoiled helical structure. You can see the cytosine, guanine, adenine, and uracil. These are the nitrogenous bases involved in RNA structure, and these are the types of RNA. As I mentioned you, majorly we study mRNA, tRNA, rRNA, the major RNAs which we study. But other than that, you also have your snRNAs, SNOs RNAs, scRNAs, satellite RNAs, a number of other, but majorly we study mRNA, then comes your tRNA and your ribosomal RNA. Your mRNA is a messenger RNA, and your tRNA is a transfer RNA, and rRNA is your ribosomal RNA. Other than that, you have your heterogeneous RNA. Now, if you see the messenger RNA, this messenger RNA basic function of mRNA is to transfer the genetic information from genes to ribosomes to synthesize protein. So the information from the DNA is being transferred from the genes to the ribosome to synthesize the protein. That is the major role of mRNA. Whereas heterogeneous nuclear RNA, this will serve as a precursor for mRNA and other RNA. Precursor in the sense a triggering agent. Okay, like it initiate a particular service. This heterogeneous nuclear RNA acts as a precursor which serves for mRNA to start the function. And transfer RNA, it transfers the amino acids to mRNA for protein synthesis, which we all know, which we study under the transfer of amino acids to mRNA for protein synthesis. And of course, you can see the ribosomal RNA. This ribosomal RNA, they provide the structural framework for ribosomes, where the construction of the protein takes place. So the small nuclear RNA, this snRNA is small nuclear RNA. This involves in mRNA processing. This table gives the overall picture of the types of RNA and their functions. It's a wonderful table. It's available online. If you can go through, you can go through again. And the small nuclear RNA, that is your small nuclear RNA, this involves in mRNA processing. Whereas small nucleolar, that is snRNA, this plays a key role in processing of your ribosomal RNA molecule. So for ribosomal RNA, it is small nucleolar. Whereas for mRNA, it is nuclear RNA. And of course, you can see small cytoplasmic RNA. This small cytoplasmic RNA, that is scRNA, involves in selection of proteins for export to your ribosomes. Okay. And you have transfer a messenger RNA, that is tmRNA. This mostly present in bacteria, and they add small peptide tags to the protein to facilitate the degradation of incorrectly synthesized protein. Like now the product is wrong. So to identify the product which is being produced in bacteria, it has to be tagged. So that tagging is done by transfer messenger RNA. Yeah. So hope you have understood about the RNAs, types of RNAs, and this basic structure of DNA as well as RNA. In the next class, I would like to continue with the replication part.